Great. Okay. So yeah, as Jesse mentioned, I'm Steve Meisburger. I'm actually a research associate now. I'm a postdoc, still in Nozomi Ando's group at Cornell. So I guess I've been doing small angle scattering since about 2008. That's when I did my first synchrotron beam time as a grad student. It's a fun technique, and I'm really pleased that you guys are all interested in learning it. So this talk will be mainly to introduce the SEC-SAXS technique to you. I'll try and teach you how to analyze data, and also at the end I'll give you some examples of more advanced things you can do with chromatography coupled SAXS and sort of what's on the horizon there, because this is a sort of rapidly developing technique. So what is SEC-SAXS? So in the upper right here, this is just showing basically all that an SEC-SAXS experiment is a chromatography system which has been fed into a SAXS flow cell. And so you're collecting X-ray data at the same time as you elute from a column. And you frame the camera continuously, and that gives you intensity as a function of Q and also as a function of time. So you'll end up with a stack of scattering curves, which you can visualize in 3D like this. That's kind of my preferred way of looking at SEC-SAXS data. And then these experiments are usually done at a synchrotron because you benefit from having high flux and small beam size, but they can also be done at a home source or even neutron scattering, as demonstrated SEC-SAXS, or SEC-SANS in that case. Okay, so I'm zooming in now on this cartoon, and I'm just going to go through what the different components are. Many of you will be familiar with size exclusion chromatography because it's kind of a standard thing that's done in protein purification, but I know some of you may have not heard of this before, so I'll try and go through what these parts are. So in a typical setup here, you'll have a buffer reservoir and a pump, and you'll flow continuously, this buffer continuously through a column. There's usually a sample storage loop and an injection valve, so you'll inject the sample using a syringe into this storage loop, and then it will be fed into the column at the appropriate time. So as your sample passes through the column, it goes usually through a UV cell or some sort of light absorption measurement, and then into the SAXS flow cell. And then, of course, the SAXS setup is exactly the same as you would use for a regular experiment. So it's just a vacuum flight tube, a beam stop, and a detector. So why would we go to the trouble of doing SEC-SAXS? There's actually a lot of advantages, and I think people maybe didn't appreciate this so much when they started, but you get an exact buffer match from SEC, which means you'll get very good buffer subtraction, and that can be really helpful. The SEC column is great for removing aggregates, which sort of are a common pitfall in SAXS, is that you have some aggregated stuff that's hard to remove. You can, by analyzing the data carefully, you can sort of confirm that your sample is monodisperse. There's really one dominant conformation or dominant scattering species. You can also do advanced analysis to separate mixtures of things. If you have, for example, monomers and dimers, you can get both of those from the same experiment. And even when peaks are overlapping, meaning you can't actually purify your protein by SEC, you can still sort of computationally purify the scattering. And I'll show some examples of that later. So that makes this really powerful. Now, there are also downsides. And, one of the downsides is that it tends to use more material. It takes longer to collect data than a corresponding static measurement. Another disadvantage is that the buffer is kind of a fixed quantity. So it's quite difficult to change buffer conditions because you have to re-equilibrate the SEC column. And that means it's hard to do things like titration experiments. And then the other issue with SEC is that as your peak is eluting, the protein concentration is changing. And so if you have a oligomer that has a weak binding constant, then that oligomer could fall apart. And then the other issue is radiation damage can be an issue because you've got the beam on constantly during the experiment. Okay, so here's just a timeline. This is showing the number of publications per year according to a Google Scholar search. this is the publications that mention SEC-SAXS. You can see it's really taken off in the last 5 years or so. And this is kind of becoming actually one of the standard techniques that everyone does now when they have a new sample to look at. I'm just going to go through on this timeline and point out a few like kind of key dates. And if you go to the very end of my talk, you all have copies of my slides. So if you go to the very end, you'll see the complete references. Sort of the first demonstration of SEC-SAXS was around 2004. And you can see it took a while before this sort of caught on. And that's partly because it had to be sort of implemented at all the beamlines around the country. And then more recently, we've got some new developments, like you can do other kinds of chromatography other than SEC. So several groups have demonstrated anion exchange coupled chromatography with SAXS. There's also a method called CoFlow that I'll talk about at the end, which can be used to reduce radiation damage. And then there have also been developments in data analysis. So kind of one of the most important techniques we use in SEC-SAXS is called singular value decomposition. And that's actually quite an old tool from linear algebra, but it's sort of first used for SAXS data in the mid-90s. And then there's also sort of analysis techniques that are really tailor-made for SEC-SAXS. And the first kind of major one to come out was US SOMO. I'm not going to talk about US SOMO, but just be aware that it exists. I'm going to talk about EFA and Regals. These are two methods that I was involved in, so they're sort of, I know them well. So Kushal yesterday actually went through the basics of size exclusion chromatography already, and so you've actually seen some of these slides before, but I'm just going to go through them quickly again to refresh your memory. So in SEC, as I've said, we have a pump and a buffer and a sample injection loop and a column. If you zoom in on that column, what you'll see is a bunch of beads which are packed in this column, and there's sort of your flow goes from top to bottom, and you have the buffer that's flowing through is referred to as the running buffer. And when you're doing size exclusion chromatography, in the lab, you'll typically have also like a conductivity sensor or other sensors in line so you know what you're looking at. Now, if you zoom in on these beads, what they're actually composed of is it's a porous, usually polymer material, or silica, and so you can have the pores in this matrix are such that large objects are excluded from the pores and small objects can go in, and so large objects that are excluded will tend to run quickly through the column, whereas small objects, which can diffuse into the pores, will run more slowly. And so on the right-hand side, this is sort of showing visually how these different red, orange, and yellow proteins would run through the column. So you'll see the red one is running more quickly through the column because it's larger. And then one thing to keep in mind when you're reading chromatograms is that the thing that goes faster elutes first. So that will typically appear on the left-hand side of the chromatogram. So here's an example of a SEC chromatogram, and on the very left-hand side is, the high molecular weight peak, and on the very right-hand side are the smallest molecular weight objects. And, in things of intermediate molecular weight fall in the middle. And exactly what these molecular weights are is a property of the column that you're using. So here's an example of this is a calibration sample run on a SuperDex 200 column. This is a pretty common column used in SEC-SAXS. And usually what people will do is they'll make a concentration plot like this. So this is K is the partition coefficient, so basically the point at which it elutes on the column, and then the x-axis is the log of molecular weight, and for this calibration sample, it falls on a straight line. So in other words, the volume at which it elutes is proportional to the log of molecular weight. Now this is true for, this sort of calibration curve is true for globular proteins, but if you have proteins that are not globular or you have things like RNA, they can deviate a lot from these calibration curves. So actually SEC is not a great method for determining molecular weight. Nonetheless, you can use these calibration curves as a way of getting a rough idea for where your protein ought to be eluting if it has the sort of molecular weight you expect. And also it can be helpful for choosing which column to use for examining your protein. That said, there are sort of lots of different parameters that go into choosing a column, and this sort of massive table on the right lists sort of the typical choices that you'll be able to make. In terms of brand of columns, most beamlines that I've found use either GE columns or Shodex columns. And the GE columns are a polymer and sugar-based bead, and they're very commonly used in protein purification, so people who do protein work will be familiar with them. And they also have a very wide range of chemical compatibility. The most important thing to keep in mind is for Shodex columns is that they can only work at pHs less than about 7.5. So if you have something that requires a basic buffer, you'll have to use a GE column. But usually this is something that actually the beamline scientist will advise you on. So there may be some columns available at the beamline, or you may need to bring your own. Okay, so this table, is mainly for your reference. Another thing to keep in mind is that for those of you who do protein purification, you might be used to using very large columns, but for SEC-SAXS, you tend to use analytical scale columns, which will be in the range of 2.4 to 24 mils volume. Okay. So here's this cartoon that I drew of the SEC-SAXS setup. So let's actually look at some pictures. This part of the chromatography setup is usually going to be an FPLC, so like a sort of integrated unit that contains all the components. This is an active purifier that is used at the Chess G1 beamline, so Richard's beamline, and I think they have sort of similar, maybe Agilent stuff at BioCAT. And then you of course have your SAXS beamline. This is again the G1 beamline. And what's done is that you just literally just put a piece of tubing from the FPLC to the SAXS beamline, and now you have chromatography coupled SAXS. So it's really quite a simple setup. An important thing to consider is the design of the flow cell that's used. So a lot of beamlines will use these glass capillary flow cells. For example, this is similar to the one that's used at Chess. I believe BioCAT uses CoFlow, but I'll defer to Jesse on that. But I think it's also based on a glass capillary. So the procedure for data collection is also pretty simple. So you collect scattering images in real time, you integrate those scattering images to get curves, and then you have this data set which can be visualized in 3D as a function of time and Q. And then you can subject this data set to further analysis. And so for this, I've kind of divided analysis into sort of a couple of sections, so basic and advanced. So basic, this is kind of what the analysis you'll want to do at a minimum. Typically, as you're collecting data, you can actually do this analysis. It's quite straightforward. And what you want to do is you'll want to make something like a chromatogram from the SAXS data. The usual thing to do is to plot scattering intensity and RG across the peaks. So I'll show you kind of how that's done. For an example data set, I'm just going to show this one that I collected many years ago from BSA. BSA is kind of an interesting standard to use for SEC-SAXS because you can just dissolve the powder in some buffer and inject it, and you get a curve with multiple bumps in it. So it's kind of nice. No fancy preparation required. So here's the chromatogram from BSA. So what you'll see is we have a main peak. This corresponds to the BSA monomer. We also have some other peaks on the left, and these are higher molecular weight species, perhaps dimers or trimers. Oh yeah, I'm sorry. This is not the scattering intensity. This is UV absorption. If you plot the scattering intensities on top of each other for this data set, this is what they look like. And you can see that this blue part, this is the protein coming off the column. This is in the elution peak. Okay, so how do we turn this into a SAXS chromatogram? So one thing you can do right away is just plot the average intensity or the sum of the intensity at overall Q at each time point. And that gives you a plot like this. So this is mean intensity versus volume or versus time or frame number. And you'll see that it looks a lot like that UV trace that I showed before. But there are some interesting differences. So for one, you'll see there's this negative going peak on the right-hand side. And that negative going peak, it turns out, is due to a buffer mismatch. So the sample that I injected had a slightly different buffer than the running buffer on the column. So that's something to keep in mind. It's not always this obvious, but often you'll have a peak on the right that corresponds to the buffer peak. In this case, it was easy to tell that this was a buffer mismatch peak because there was also a bump in the conductivity trace. That's this red trace here. So I think I had no salt maybe in my BSA buffer and there was salt in the running buffer, something like that. So the reason this is important is that you want to choose a region of your data to use for buffer subtraction. And typically what we'll want to do is choose the region before the peak, because this will be unaffected by the buffer mismatch issue. And I guess there are certain situations where you'll have to do something more fancy with the buffer, but sort of the best practice is to do this. So now that you've chosen a buffer region, you can do buffer subtraction at each point, and then you can do Guigné analysis, in this case automatically, for every scattering curve in your data set. And now we've got a plot of forward scattering, I0, and the RG in purple that are both determined from Guigné analysis. And now you can see that this bump on the left, corresponds to something with a larger RG, and the RG is fairly constant across this peak region, so it's probably a single component coming off. Okay, so at this point, often this is where people run into problems. So not everyone's data set will look nice like this and clean. There will be lots of possible issues. And so this is the point where you will do some troubleshooting. I'm not going to go through all of this. This table is mainly for your reference, but I'll just highlight a few common issues that you can have with SEC-SAXS. So one is that the SAXS profile does not return to the baseline after the peak. In other words, if you look at the buffer curves before and the buffer curves after, they look different. And there are various reasons for that. I mean, it could be that radiation damage occurred and caused protein to stick to the X-ray windows of your capillary. Or it could be that you hadn't fully equilibrated the column before running the protein. The other thing that can happen is you could have RG not constant across your peak. And that may be caused by interparticle interference if your protein concentration is very high. Or it could be that you have overlapping peaks. So there's actually multiple different species eluting that are in sort of an overlapping region. And yeah, these overlapping peaks can be a real problem. It could be that your column resolution is insufficient, or that you have something that's causing the peaks to broaden out and overlap. Or you could have like reequilibration of monomers and dimers on this time scale of your experiment, which can really mess with things. So these are all things that can happen, and you may need to adjust things with your experiment in order to fix them. Okay. Now, some of these issues can also be addressed computationally using more advanced analysis, and I'll talk about that next. Oh, before I do that, though, I wanted to give an example of what this radiation damage can look like. This is an example from a really nice review by Perez and Vachette, which I recommend you all look at. And this is like an extreme example of radiation damage. So in this case, they had a protein that in Heaps buffer was very well behaved, but in phosphate buffer would damage very easily in the X-ray beam. And so in the Heaps buffer, that's this red and orange trace. In the Heaps buffer, it's very well behaved, so you can see it's quite a flat RG across the peak, and it goes back to baseline. But in the phosphate buffer, I of 0 or RG just goes up and up and up and up and up over time and never comes back down, right? This is definitely bad. And so this is the sort of thing to look out for. It's usually not this obvious, but sometimes it can be a problem. Okay. But before I talk about advanced analysis, I'm going to introduce singular value decomposition. This is kind of SVD is kind of the most basic mathematical technique that sort of underpins all the other methods I'll talk about. So I'm going to spend some time on it. So the reason that we can use SVD is that In very dilute solutions, the theory of SAXS tells us that intensities just add. So I'm borrowing this cartoon from Kuchal, or this metaphor of fish and sharks swimming together. So if the fish and the sharks don't interact, then the scattering from fish and sharks just adds together. And so you end up with this purple curve on the left, which is just the sum of the yellow and the green curves from the respective components. So because these things are additive, we can use methods from linear algebra to deconvolve them. And the method that I'll talk about is SVD. This was first used for SAXS, I think in 96 or 95. And basically, what you do is you have a matrix A, which contains your data set. So the SAXS profiles are arranged as columns in this matrix. And you can decompose it as the product of three matrices. And these matrices are U, S, and V. The S matrix is diagonal, and it contains positive numbers along the diagonal, which are called singular values. And the U and V matrices contain, so U contains scattering profiles along the columns, and V contains sort of concentration peaks on the columns. So that's really cool because you can take your data set, which is a mixture, and separate it into sort of components which are effectively, additive components in your data. And if all goes well, the number of singular values, which is above this sort of constant level, is telling you how many different components there are in your data or in your mixture. So in this case, you see three components, or three sort of significant singular values. You might say, oh, there's probably three components in my scattering. Okay. So this should tell you that you can use SVD. Since you can use SVD to detect the number of significant components, you can sort of identify which parts of your chromatogram can be averaged together to get a single scattering curve. And this is just kind of a fun example you can do. So for my BSA data here, if I select the whole curve, so from the beginning to the end, and I plot the singular values, you can see three significant ones above this noise level. If I now exclude the negative going buffer mismatch peak, now you can see there's really just two significant components, as you'd expect. Now, I think I said that the shoulder here is probably a dimer or trimer, so we can exclude that shoulder, and now we see only one singular value. So that's telling us that all of the data in this range can be averaged together into a single scattering curve without really losing any information. And SVD actually does that average for you. It's just the first column of U. Although if you look at the first column of U, the first thing you'll notice that in this case it's negative, which is wrong. You can't have negative scattering. So it turns out SVD doesn't know about the sign of scattering. It could be positive or negative, and it's ambiguous. But all you have to do in this case is flip it, and you get scattering curve of PSA monomer. Okay, so now you might be wondering, well, what if I have a really complicated data set? What would SVD tell me about that? So here's another fun test case. This is, I just loaded up a whole bunch of standard proteins into a mixture and injected them. This is the BioCAT chromatography standard, which is, a bunch of proteins and vitamin B12, and you get a chromatogram with a bunch of peaks in it. So if you do SVD on that, now you get at least 7 significant components, and here's what the basis vectors look like. So columns of V, these are the things that are like concentrations. Columns of U are the things that are like scattering curves. And what you see immediately is that this can't be physical because you have concentrations which are negative, and you also have some regions of these scattering profiles which go below 0. So that can't be right. So, we've got multiple peaks in V, you've got negative scattering. What's wrong? So this is one issue with SVD, is that typically on a complicated data set, if you do SVD, the components themselves will be non-physical. But you can, the key thing is that to get physical components, they're just going to be linear combinations of these guys. So I'll show you a method in a little bit that can sort of do that, those linear combinations automatically. Now to get a little more insight into what's going on with this method, I'm going to show you something else. So this is what would happen if you did SVD in real time as your data was coming in. So on the left, I'm showing sort of scattering curves being plotted in real time, and so now you see some protein peaks coming out. And then on the right is the spectrum of singular values from an SVD of that data set. And you can see that as proteins pop out, you also see new singular values popping up above the noise. And so SVD, as you can see, is a really sensitive way of detecting when something first elutes. Using SVD in this way is called evolving factor analysis. It's actually kind of an old technique from the 80s. And when you make a plot of those singular values versus frame number, it's called a plot of the forward evolving factors. And so from this plot, you can see that it's easy to identify when new components elute based on these sort of strong inflection points in the singular values where they pop up above the noise level. You can also do the same thing in reverse. So in other words, as if you had collected the data backward in time. And this type of plot tells you when components leave the scattering volume. So now you have a way of telling when components enter the scattering volume and when components leave. And from that, you can make little windows for each peak. So now you know where each peak starts and ends. And this is done using a sort of first in, first out principle for SEC. Okay. And then the final step, once you have the windows, you can use those windows as a restraint to do a linear least squares fit. And you can basically find linear combinations of these guys that satisfy those restraints. You know, the restraint being that the concentration is non-zero only within the window. And from that, you can get peaks and scattering profiles for each thing. So that's the EFA method. You can do, so both EFA and SVD you can do in raw. So this is, on the left is the SVD window that you'll see, and on the right is the EFA window. And you can also read more about this in Jesse's paper on raw from 2017. Okay, so for the last part of my talk, I'm just going to give you a sort of a taste of some more advanced applications and methods. So I've shown you how you can do SEC-SAXS. Well, could you use other kinds of chromatography? This is an example of anion exchange chromatography. So in AEX-SAXS, what you're doing is you have a strongly charged media, and then the protein, which is oppositely charged, will bind to that media. And then you can elute the protein by using a gradient of salt, for example. So at high salt, the protein will elute. And you can use this technique to separate based on sort of surface charge properties of your protein. Now, our collaborators had collected data on this ribonucleotide reductase enzyme from B. subtilis, and this is the AEX-SAXS, or the regular AEX data that they collected. So this dashed line is showing the NaCl gradient, and you can see that there's sort of multiple peaks coming off of this column, and they determined that these different peaks corresponded to different ligand states of the enzyme. So it was co-purifying with some ligands, and those are labeled holo and apo. So they wanted to know what those states were structurally, so we decided to try AEX-SAXS, and this is the sort of data set we got, and you can also see that there's a couple peaks. Now, analyzing this data is tricky because you have a sloping baseline from that salt gradient. And we actually came up with a new method to do that called Regals, and I'll talk about that a little bit later. By using the Regals and to do the deconvolution, and then by comparing the SAXS data with different models we got from crystallography, we're able to determine what the structures were for holo and apo, or to assign those to high-resolution structures. So another thing that's cool you can do with SEC-SAXS is you can look at membrane proteins. So often people will use detergent to solubilize membrane proteins for doing analysis like SAXS, but this can cause problems because, first of all, the detergent corona around the protein contributes scattering, which is quite a lot. But the other thing that at least I didn't appreciate at the beginning is that when you're using high concentrations of protein for SAXS, you're often going to be above the critical micelle concentration. So that means that free micelles will be forming spontaneously in solution, and it's very hard to control that. So in this case, what was done is they used aquaporin as kind of a standard protein that's a standard membrane protein solubilized with EDM, and they used SEC-SAXS to equilibrate that protein with a buffer containing detergent so that they had a good buffer match that contained free micelles to the protein plus free micelles. In other words, zone 1 here, which is just the elution buffer, and zone 3, which is solubilized protein plus free micelle. They could just subtract those two and get the protein alone. That combined with atomistic modeling of this detergent corona, they were able to get good fits to the scattering profile. Okay, so I've mentioned this co-flow system a couple of times. And, as I said, one of the issues with SEC-SAXS can be capillary fouling or radiation damage, which is a problem particularly at high flux beamlines. And this can compromise data quality for sure. So this co-flow system is a way of getting around that. And what it does is it focuses the protein down into a sort of jet with a sheath of buffer around it. So the protein never touches the capillary wall. But just by doing that, it seems to almost completely eliminate the capillary fouling issue. And so you can collect data with much higher X-ray flux without having to worry about the damage. And I believe BioCAT has this now as their standard mode for SEC-SAXS, although
we've
Jesse
been running the co-flow standard for about 2 years now. So
yeah, great. So you won't have to worry about capillary fouling at BioCAT, hopefully. All right. The other thing, and this is also implemented at BioCAT, is that they can do multi-angle light scattering in line with SAXS data collection. So Kushal, I think, talked about mols yesterday as a technique for getting molecular weight on an absolute scale. And that's sort of what's shown here, is that you can get sort of very accurate molecular weights by doing multi-angle light scattering in line with SEC. And so it's sort of natural to consider doing SEC-SAXS and mols at the same time. This is the Sec-sax mols setup at BioCAT, where you can see the chromatography system, and then these blue boxes are the mols and DLS refractive index instruments from Wyatt. And so by doing this, you can get, sort of a whole suite of information during your experiment. Now, one thing to keep in mind is that for doing mols well, it requires sort of more care with sample preparation, choice of column media, equilibration of columns. These things are all somewhat more difficult than just SEC-SAXS. So yeah, keep that in mind. The other thing to mention is that this is also now available at many places, not only BioCAT, but also Chess, ALS, Petra 3, and Diamond have similar setups. Okay, and finally, I was going to talk about Regals. This is this method we came up with for analyzing chromatography-coupled SAXS data where you have sort of non-ideal situation. So I think I may have, so EFA can fail if the peaks are highly overlapping or if the background is changing, as in AEX-SAXS. So what we do with Regals modeling is we add new peak and concentration models that help us deconvolve components. So these models are basically, for the concentrations, they can be sort of smooth, arbitrary peak shapes. And for the SAXS profiles, they can also be smooth, arbitrary profile shapes, or they can be P of R functions. So in other words, you could say one of my protein peaks has to have a Dmax of 100, and use that as a restraint for doing your deconvolution. The first example of this was to analyze the AEX-SAXS data set, where the constraint we imposed was that the buffer scattering components had to be very smooth. So they couldn't have any bumps in them. They had to be very smooth lines. And just with that restraint alone, we're able to deconvolve the data set. So we didn't have to restrain the SAXS profiles at all. One of the cool things about the Regals method is that it's very general, and you can use it on other kinds of data, including time-resolved SAXS and equilibrium titrations. And we sort of recently published this in IUCRJ, and actually all of our code and our example data sets are online, so you can play with these yourself if you want. I should also say Jesse has been working hard on a GUI in raw, so you can do Regals without having to write any code. So stay tuned for that. Okay, so to summarize, SEC is a way of separating molecules based on size. And SEC-SAXS is a great combination because it lets you remove the aggregates, separate oligomers, provide a good buffer match, and just increase your overall confidence that you have nice monodispersed protein. It typically requires some extra sample and time, but often that's worth it. And you have to take some care to do SEC-SAXS well. So be aware of the different experimental variables that affect data quality, and also, check your data quality, ideally as you're collecting data so you can fix problems. And then, one of the advantages is that we have all these powerful deconvolution methods like SVD, EFA, and Regals to get sort of more out of your data than just averaging profiles together. And there are also many exciting technical developments underway. So it's a fun field to keep track of. So I'm happy to take questions now. I also have a few slides after this one which are for your reference, and I'll pop those up. Oh, I also want to mention that I'm a member of the Ando group, and I showed some of their data. These are the grad students, postdocs, and this is Nozomi Ando here. Check us out on Twitter, and we have a GitHub. Okay, so here is, this is just a sort of exhaustive table of SEC-SAXS capabilities at various user facilities in the US, at least according to the most recent information I could find online. So as you can see, a number of beamlines in the US have SEC-SAXS, and many of them have other capabilities as well. For example, Chess can do high pressure SEC-SAXS, which is sort of a unique capability. BioCAT has Sec-sax mols and CoFlow. And then there are also many facilities outside the US. Importantly, SEC-SAXS is a great technique for doing mail-in because you don't have to worry so much about your buffer matches and all that because that's taken care of by the column. And many of these facilities allow mail-in as well. So yeah, lots of options. This is a list of references that I sort of referred to in my slides. And also, I really recommend this review by Perez and Bichette for more detail